Doke. Yeah, it's all working. Back to our notes. Okay. It's gonna do me dirty again. Let's just do it live, I guess. Okay. So for our discussions on course, right? For our discussions on research methods, we've been talking about the different sort of stages or classes along the progress of the life cycle of research. And we've taken a winding road through them because there are lots and lots of good questions we've been addressing along the way. So I'd like very much to continue that sense of let's ask questions as they come up. We're moving through this fast enough, so don't feel like you guys are derailing the class. Let's continue on with more of the sort of question and answer discussion format versus just the knowledge transfer of lecture. Okay. So we're up to talking about explanatory research, and we've been throwing around terms like necessary and sufficient causes, contributory causes, and what makes research clearly descriptive, putting in a bunch of adjectives around the phenomenon that you're studying and figuring out the intensity or list of those adjectives or descriptors or characteristics or attributes of the phenomenon versus looking at what drives the phenomenon, what gives rise to it, how does it evolve through time or space, or how does any set of inputs to this phenomenon, how does it become the outputs that is characteristic or that are characteristic of this phenomenon, right? So mechanistic modeling. And explanatory research is where we are building different versions of that mechanism that the phenomenon represents and seeing how to break them. Because if we put in inputs to some black box that we don't understand how it works, and we get out results, we haven't learned anything about the inside of that black box, right? It's still an opaque box to us, still an unknown mechanism. But if we play around with different inputs, some of which seem to cause the black box mechanism not to work, then we can start making some pretty educated inferences, start building our covering law or theory about how that thing works, right? If you regard a human being as or graduate student as a black box mechanism for producing research, journal articles eventually, stuff like that, your thesis, dissertation. As it turns out, when you don't include air as an input, you don't get any journal articles out because, as we know from the mechanism of how graduate students work, suffocation is really hard on the publication process, right? So we would never do that kind of an experiment, but as sort of an absurd conceptual example, we get the sense of we play around with the inputs to the system to figure out how the system works and to see how outputs are affected. And we call each of those trials an experiment. Okay, so we want to find new mechanisms. Typically, in the past century or so, this means of finding new mechanisms is either looking smaller in more detail at something. So going from the, in the mid to late 1800s, sort of the germ theory of disease, and people like Joseph Lister, the namesake of Listerine, figuring out that these little invisible things to the naked eye called germs cause illnesses. And what does that mean for safe drinking water and so forth, and cholera outbreaks in the UK in particular? So Joseph Lister, a guy named Scope, right? If you've ever used Scope mouthwash, that's why it's called that, right? They're trying to kill bugs in order to make your mouth a safer environment and have less disease associated with that. So we get to, in the past century, start to drill down on novel theories of things and increasingly test competing systems. Now we can do absolutely crazy stuff with microorganisms, right? We test drugs on specifically cultured and typed out, very well understood classes of organisms because we were looking at them at the ecological level first. There's something living in our water that's making people sick, down to the organism level of, some of these organisms seem to be single cellular. Some of them are higher order parasites like nematodes, worms. Some of them are, flagellate or spirogyra or move around with cilia. Others move in a pseudopod type way like amoeba and so forth. And then we get into the subcellular realm of looking at organelles, specific components that certain bacteria may or may not have, and down to the virus level, which is just barely larger than looking at molecules, right? A virus under an electron microscope is basically a fairly sophisticated molecular shape that delivers a genetic payload, basically, and causes self-replication. And now we're looking at the molecular level of organisms to check how specific drugs or antibiotics, for example, can either destroy the cell membrane like soap does. You wash your hands with soap, you don't need triclosan or triclocarban or other antibacterial ingredients in soap. The soap itself obliterates the entire physical structure of single-celled, non-higher order organisms, bacteria, but not protozoans, for example, which are able to survive stuff like that. But we get into things like crafted molecules such as antibiotics, right? So how do we get specific molecules through that cell membrane for organisms that have a little bit of resistance to basic attacks like soap? How do we get a molecule inside that blows apart the DNA, or chelates, it wraps itself onto and gloms onto some key organelle structure of that single cell organism and just causes it to not be able to move or process energy or whatever, and thereby kill it off and stop the disease process that way. And of course, below that, we are looking at the atomic level. So we're zooming in continuously as a process of advancing science as we're able to cybernetically extend our senses with sensors like electron microscopes to figure out what is the shape of the SARS-CoV-2, the coronavirus. How does its protein sheath change or mutate over time so that we can craft very specific mRNA sequences to instruct human bodies to create antibodies for this and get after it. I mean, just incredibly sophisticated stuff in about a century and a half now, versus even thinking that germs are a thing in the first place, right? So we are, in that case, testing competing ideas of what are these little things? Are they little animals? Was the first theory. We extend these days existing senses of causality now that we know that we have a covering, or we have developed a covering law of pathogens are similarly living or living like organisms in the case of viruses or prions, and their metabolic byproducts or their feeding or reproduction processes cause human pathologies, right? And so now we're not really trying to, not a lot of people are trying to refute that sense of germs cause illness in specific ways with metabolic byproducts, for example. Like the E. coli in your gut. Every once in a while, somebody eats some spinach that's been washed too many times with, you know, cattle droppings to fertilize it. And the poop of those E. coli is very irritating to our digestive tracts and can cause, death or serious discomfort as a function of uncontrollable diarrhea. And that's pretty well understood these days. I'm not aware of any sort of credible competing theories on that, but we can fine tune our drugs to go after those specific E. coli in the gut of somebody who's been infected with them and not kill all the other E. coli, which are perfectly good, right? So there's more bacterial DNA in your body than there is you DNA, right? You hear stuff like the gut flora and the brain gut axis and all this stuff, and how that may have all sorts of deep reaching things like causing certain neurological conditions out of the digestive tract. I mean, just amazing stuff that we're able to start fine-tuning our understanding on by extending existing causality models about all this stuff we're finding in microbiology and human health. Okay, so let's zoom out a little bit instead. The other direction is to look further out. Astronomy, for example, we're doing stuff out at the edge of the created or Big Bang universe, whatever, these days with background microwave imaging. I mean, just amazing stuff. Looking at the edge of the universe. That, if that doesn't blow your mind, nothing will, right? So let's take kind of a meso example in between looking at the edge of the universe and down to something the size of a prion or a virus. Let's ask a typical, forestry or ag related question. We have some idea that the presence of tree species, in this case, we'll use balsam fir. The presence of that organism is not just a truth or absence fact of that ecosystem. It affects that ecosystem dynamically by providing habitat, by consuming resources, sunlight, water, available temperature, and so forth. And some species, like eucalyptus, do allelopathic attacks on similar competitive species around them. They'll actually poison the soil in some radius from each individual root ball of eucalyptus against other species that would look to consume that eucalyptus's available resources in terms of soil nutrients. So the question becomes then, does balsam fir have any sort of effect on soil character around it? And the obvious qualitative answer is, yeah, sure. I mean, the presence of anything automatically affects everything around it. Like you and I can't walk through the forest without affecting that forest by scaring wildlife away briefly, by creating sound, by trampling things, right? Lots and lots of probably small, but real effects on the world around us and around every other individual organism as well. So the descriptive phase of asking what kinds of effects do balsam fir have on understory, so other not top canopy tree species, shrub species, ground cover, herbaceous stuff, so non-woody vegetation. There's got to be some effect. What would that be? All right, so the descriptive phase would ask some of those things. Does it, I mean, it's obviously adding balsam fir to the composition species list present by being there for the first time, maybe. But does it force other things out? Does it actively poison other things out? Does it synergize with any other species? And so when you add balsam fir to the equation, you also get enriched other associated species. So commensalist species, symbiotic species, stuff like that. The descriptive phase would also be asking questions of, okay, how beyond the addition or loss of other species, what happens within each of those species? Do they benefit? Do they struggle with? Are they extirpated by? Are they outcompeted by balsam fir? Do we get altered understory vigor across any of those other species that we're looking at? Do wildlife use the area, not just the balsam fir itself, but the whole area, if we're changing understory structure and function and the related species nearby? How does that change the picture for wildlife? Yeah. So wildlife affect understory as well. Wildlife affect soil quality. For example, there's a really interesting study a while back on forest soil nutrient budgeting in southern coastal Alaska, where it's so cold that the nutrient cycling, like the soil bacteria and stuff like that, they got a matter of weeks to a couple of months of frozen or unfrozen soil. And so there's just not a lot of bacteriological or fungal action cycling soil nutrients. And so grizzly bears, which are catching protein-rich salmon, and the salmon have turned a bunch of lower trophic stuff into salmon protein and oils. The grizzly bears eat those, they go up into the woods and they poop. And the primary resource nutrient input for that entire forest type in that particular area, because it's so cold, it's such a high latitude area, that's it for fertilization. So if you mess up the balance or the behavior of bears, mess up their diets, it has centuries worth of crack-on effects on the entire forest around them, because you pull away those nutrients, and with every year's freeze thaw washing nutrients out of that very low biological activity soil, you can have like tropical rainforest level depletion of resources, which we expect with very hot rain washing stuff out constantly in a very cold subarctic environment. So that really underscored one of the powerful effects of grizzly bear management. You're managing the whole forest by managing that essentially keystone species, right? But in this case, what's the opposite direction? Do the balsam fir affect grizzly bears in that particular habitat combination, and so forth? So this is all just kind of the what's, the how much, what are the wheres, which species, right? So the who, what, where, when questions. And on the how and why, how would we design an experiment to figure out how balsam fir affects. Let's just do understory. So species composition. How would you design an experiment to test your descriptive level understanding of what balsam fir are doing to the world around them? What kind of an experiment would you propose?
Plant a balsam fir into a new environment.
Hell yeah, you would. Yeah. And then you would track what happens. And you would also track what's happening at an area where you didn't plant any new balsam fir. Control. Negative control. Okay. So that's a pretty straightforward thing, right? You're asking, how can I isolate from all the other things that could be affecting the understory vigor, understory composition, et cetera, et cetera, et cetera. All those things could possibly be changing. How do I isolate just the change that I'm interested in and keeping everything else constant, right? So statistically controlling for the effects of other things. And this is field environmental research. So let's say we run this trial once a year for 5 growing seasons. So we repeat this 5 times. And time 1, we have normal rainfall. Time 2, we have low rainfall. It's an El Nino or La Nina, and in that particular area, that means reduced rainfall. And then the 3rd year, we have high rainfall, and so on down the list through our 5 years. Because we have experimental plots and a control, it doesn't matter that the rainfall level has changed, because we can compare and say, well, you know, the rainfall lack also happened on our control plot, half a mile over from our experimental plot of a whole bunch of planted balsam fir, and we can subtract the effect of missing rainfall or drought-like conditions from our understanding of the understory vigor. All of the plants suffered that summer. So if we see that the fir plot understory vigor was 39% of expected in our treatment area versus 42% in our control area, then if we know that the rainfall approximately affected both of those plots equally, we would guess that the balsam fir had about how much effect on understory vigor? 3%, exactly, right? It can be that simple. Now, it's never actually that simple, right? So we have, you know, we'll get into experimental design and stuff like that, and there are entire classes just about experimental design for the PhD students. But, you know, the idea is we want to have controls for features or factors that are pushing our dependent variables, our response variables, our outputs up or down, but that are not the point of our experiment. We can still control for that. Not necessarily using a control, but for example, by using blocking. So let's say in our descriptive phase of research, we find that balsam fir at the top of a slope have a huge effect on everything around them because top slopes are exposed. They're getting hammered by weather real hard. It's a more difficult growing condition. And balsam fir, as evergreens, can sort of hang on and be at least minimally biologically productive right through the winter, whereas some leaf-off species have to go dormant during that time. Some deciduous species. So we would block that experiment for our treatment area and our control area. We'd subdivide those into ridge, mid-slope, and valley strips, right? So if our treatment area is like a big square on the map, and our control area is a similarly big square on the map, we stripe that, we place those squares such that there's an even ridge section, a mid-slope section, and a valley section, and make sure that those are reflective and approximately proportional in sub area between treatment and control. And then we have controlled for the known effect of landform position, ridge, mid-slope, or valley, because we know that this has a real effect on our data. So if we don't block this out statistically by having these all present and accounted for as another input variable, well, then we might accidentally put our entire treatment plot on a ridgetop, and we might put our entire control plot anywhere but ridgetop on accident, or maybe 75% ridgetop, 25% ridgetop. But they're both the same size, so on paper, without looking at the map, you'd say, all right, this is a good control and a good treatment. They're well paired, but they may not be. So for a lot of kinds of field research, we're going to have one or several statistical blocking factors like this that you have to account for. Otherwise, you know, if this is our treatment area is on top of that ridge line, and we're not accounting for that ridge line effect, remember, balsam fir could do really pretty well on a ridge line in this totally hypothetical example. It's not actually true, but in our hypothetical, we'll go with it. So we see this effect here in our treatment of the unblocked area, when we're not taking this into account. We see a 31% effect, and Cx is constant at 42. So here our conclusion would be, oh my gosh, there's an 11% effect here, and somebody else did a similar study and they only found a 3%. We got almost 4 times the effect that some other schlubs did. Our research is better. It's actually worse because we're not taking into account the 8 or 9% effect, let's say, of landform position, right? So statistics give you the ability to block this out. In qualitative research, we're asking questions and having conversations along these lines, but we're not mapping things onto numbers. We're looking for, they're called pithy quotes, that 2-sentence answer to one of your questions out of that whole hour-long conversation that really captured the beating heart of, oh, okay, you know, it seems like some folks are wrestling with this particular problem in society, maybe redlining by a bank when they're trying to get a mortgage and they're encountering difficulties based on their perceived race or ethnicity. And the banks are low-key doing some racist stuff, whatever. Redlining has been a long thing in history and is super duper illegal now, but can still pop up here and there indirectly. You're looking for that quote of, man, I don't know what's going on, but we took our credit reports around to 5 different banks around town, and the only one where they even would talk to us was this bank here. And so as the researcher, you think, that's interesting. That sounds like a signal to me. What's different about that bank? Because the people taking their loan application to these 5 different banks, people haven't changed, right? So they're a constant factor. We don't have to block for them. Well, we are looking for what's the descriptive, what's the qualitative difference on that bank versus the others? Oh, do a little bit of digging on that bank. They got caught by the federal government doing redlining in the past, and so they were, you know, forced by the Supreme Court to go through a whole bunch of regulation and auditing and stuff like that. And now they are super careful about, you know, being fair and equitable in loan access. And the other banks were not forced to do that. They haven't been caught yet. It could be, right? So that happens in the business world all the time, all the time. Some companies build this stuff in, those fines they get from governments, as a cost of doing business, and they keep right on doing it, right? But the way that you get that signal is looking for that, that's weird. You went to 5 different banks and got the same treatment from 4 and very different treatment from the 5th. What happened? What's different about that bank? That's weird. That doesn't make sense that one would be wildly different than the others. Banks are usually pretty similar, right? They're very competitive with each other. So, qualitative research, we're still having to think about stuff like blocking and not controls in an experimental design sense, but what's the comparison contrast? The one interesting bank versus the 4 uniform banks and so forth. All right. So it's the same kind of mindset of looking for what exposes mechanisms. Why is that bank different than the others? Oh yeah, they got caught. Okay. So the explanatory phase through careful experimentation or rich data collection, if it's a qualitative approach, gets us to being able to answer these questions. So you take that insight about the banks and you look for folks trying to get loans in some other city with different banks, some of which have been caught redlining and others which have not. And you start asking the same questions and seeing if the same thing holds true. And if your answers in the other city, the banks that got caught redlining are still denying people loans or whatever, then we have figured out how to break our idea that getting caught leads to a different behavior, a different experience for folks trying to get loans, and so forth. Okay. So the explanatory phase is trying to break our theories of things, trying to find what works on saying the null hypothesis doesn't work here. Okay. Cool. So in order to do that, here are the things that we got to set up. Temporal order. We have to have the input occurring before the output for that input to be able to possibly cause that output, right? So A must occur before B, or A cannot cause B, at least in our limited 3.5 dimensional understanding as human beings who can move forward linearly through time, but not backward. Okay, that is the central conceit of most science fiction that deals with time travel. Right, so we violate this assumption of causality of the thing. Now we also need to design our experiments in order to capture the sort of shadow face of temporal order, which is the logical fallacy of post hoc ergo propter hoc. After the fact, therefore, due to the fact. A happened, and then B happened, so B must have been because of A. That's a logical fallacy, right? Just for something happening before something else doesn't mean that the earlier something caused something else. That is post hoc ergo propter hoc. All right, cool. We're also looking for this idea of association. So we're looking for, it's got to have correlation present. And correlation is usually more of a descriptive level thing. But we're looking for those correlations in descriptive research to be able to set up our questions of cause and effect, causality, temporal order, association, etc. If there's no association, it doesn't matter if something happens before something else. If these things behave completely independently of each other, there's no association, and therefore A is not influencing or causing B. It just, there's no observable relationship between them, no association. We are having to keep in mind, of course, exceptional cases where in rare disease research, you're not looking for the center of mass of the 10,000 people who don't have a rare genetic disorder. You're looking for a 1 in 10,000 or 1 in 10 million that does. And so The statistics that we use to establish things like association, those are looking at the center of mass. They're looking at the 10,000. And so if you are looking for the central tendency of a large population, you're going to miss those exceptional cases. And you're going to be using a pretty different statistical approach versus somebody like an epidemiologist, for example, who is looking specifically for those exceptional cases, those rare ones, and trying to find some kind of a disease spread mechanism or disease pathology, how the damage happens as a function of the disease, and so forth. Okay. So association, just like with temporal order, it's got kind of two faces to it. With temporal order, we are looking for A before B, but we are looking to disprove just the idea that A happened before B, therefore B must have happened because of A. With association, we're either looking for what's the center of mass, the central tendency values, the average, mean, median, mode, trimean, harmonic mean, geometric mean, arithmetic mean, whatever. All different ways to say what's the middle of this thing, where's the belly button on this huge point cloud, versus an epidemiologist or qualitative case study approach where there are millions of businesses in the United States, but there's one Apple, right? So if you want to learn how Apple does their thing, you study Apple and you ignore the other 1 million. Okay, cool. And then with association, we're also looking for this aspect of causality, right? So necessary versus sufficient, like we talked about on Thursday, where if it is a necessary association, and so A is associated with B, and without A, there is no B, and A occurs before B, well then that's a fairly detailed, robust mechanism of the understanding of A's effect on or cause of B. But if it's sufficient, as it often is with field research, and especially research with humans, so we are complex and illogical, you run into the case of, man, like 22% of my human respondents are testing out where they have this causal factor in whatever you're studying with them. And like the other 70+% don't. But the other 70% are also showing some of the output behavior, but not the input behavior of the clear A goes to B, input to output. So A is, in that case, a sufficient causal factor or association with the output of B. There must be something else, because my theory about A leading to B in human beings explains 20, 22%, 25%, might only be 5 or 10% of the population. Clearly, there's some other factor like A, but that is not A, that leads to B in this human population. So those are a sufficient, not a necessary cause kind of situation. So that's very common. And statistics like the R squared thing, you have heard of R squared? This is usually a first or second stats class level statistic where, again, in a point cloud of input factor A and output factor B, B is on the y-axis or vertical axis, we get this point cloud of association. And the point cloud has some typical shape to it. Sometimes it's a conic shape. So the error in A is proportional to the error in B, and it grows like this. Or sometimes it's more of a straight line or not growing point cloud. So the error in A does not scale with the error in B, and that's helpful statistically. Well, we try to draw that line through the middle. Usually a straight line, because that's simpler and easier to think about. It might be a non-straight line, like an exponential, or an exponential parabolic, or hyperbolic, or it might be a sigmoidal S shape along this point cloud, or whatever the actual relationship is. It could be an oscillator or multimodal. Whatever. The R squared is the measure of, or the summary statistic of what's the general distance, what's the most likely distance from each of these individual points to the line, the best fit or least square error fit line. And another way to interpret that R squared statistic is if you multiply R by 100, R might be expressed as a 0.32. You would say that A is responsible for, if you can check all the other boxes on causality, A is responsible for 32% of the behavior of B. Okay, very, very, very helpful and straightforward to interpret statistic in R squared. Okay. Now, with the very common muddy situation of we have multiple competing sufficient causes, complementary or stacking sufficient causes, right? We want to make sure that we have a real clear, or we are trying to develop a very clear understanding of what are some of the alternatives that I haven't accounted for, right? So if I'm noticing a whole lot of skin cancer incidents in people in Australia, so we'll adapt this out of some real world epidemiology research. If since about 1980, skin cancer rates in Australia have gone into orbit, just super high. Lots of people getting sick, lots of people getting harmed by having skin cancer, which skin is the largest organ on your body, and cancer anywhere in your skin makes it pretty easy to move that cancer, shedding cancerous cells anywhere else on your body. It can be quite fatal. We could have some ideas of what causes skin cancer. Why is that such an issue in Australia since the 1980s? So let's throw out some ideas. We got about 8-9 minutes of class left. What could that possibly be? Location from the equator. Okay, so. And by location, you mean latitude specifically? Okay. So high latitude means closer to the poles than to the equator. And that means, okay. So physical radius from the sun. Yeah. Good. We're narrowing down a little bit. This is an idea called parsimony. The more specific is more valuable and useful and harder to show than the more general, the vague, the looser idea. Okay, so whenever we start putting together a mental model of things that you'd like to or potentially would be able to test, you want to start with the general idea and narrow down as much as you possibly can. Parsimony. Cool. All right. And the other half of parsimony is we're looking for the shortest list of highly contributory or necessary factors. All right. So if distance from the sun intensity, is the primary driving factor, then populations around the world that live along the equator, essentially in northern Africa, and like, does India cross, Indian subcontinent cross the equator? Maybe not. South America. All right. Those folks should have proportionally and equally high cancer rates as well.
En du håller en ozonlayer.
Say again?
Håller en ozone layer.
All right. Ozone hole. Okay. Good. What specifically about the ozone hole? Let's narrow that one down.
Ozone is a reflection of radiation. And we have a hole of more sun radiation reaches up without the reflection.
Destructive photolysis of atmospheric ozone, is what I heard you say. It might have been an
Yeah,
accent thing.
yes.
Yeah. Is that about?
What?
Yes. Go with it. All right. Cool.
Okay.
So, anybody in Tierra del Fuego, the very southern tip of Argentina, which is like not a lot of miles from the Antarctic shelf, they should also, as they are at times under this as well, they should be, or they could be getting cancer at just as high a rate. They are not. Interesting. Okay. Cool. There must be more to it. There must be more, at least contributory factors, whether or not they are sufficient by themselves or required. Must be more to the story. What else? Let's keep thinking.
Be like a lack of sunscreen, either like not putting enough on, or it might be like some,
Okay.
Political reasons, people thinking they don't need sunscreen. Like there's some, some quack or some influencers telling them like sunscreen is terrible because like
Social standard.
yes, yeah, like social related issues, I guess.
Or just some other lotion.
Yeah, look at that dork over there wearing that SPF 150 zinc mask. What a loser, right? You can see him from orbit. He's so white. Okay. But he's not the one dying. Okay. So social norms are a part of it. And as it turns out, that's a big part of it for Australians. Sun culture, such a huge thing around the beaches there. This is also a geography and more specifically topography, topographic effect of the fact that the outback, the great interior desert of the Australian continent, which is like what, 85 or 90% of the total land mass, almost nobody lives there. And they certainly don't have a lot of fun like baking in the sun all day long. And so the skin cancer rates were specifically associated with beach cities around the edge of Australia, not in the interior. And the skin cancer rates were also not, in some cases, present among some of the Aboriginal groups because the ozone hole doesn't equally overlap all of Australia. And there is a genetic component to skin cancer. Some folks are more vulnerable to it than others. And some people couldn't afford to go to a dermatologist, and they died a lot more because they couldn't afford it. It's complex, right? None of these things by itself is a guarantee or guaranteed protection from. But any few of them in combination is sufficient to drive that up in any particular person and multiply it out across the tens of millions of people living in Australia. We start to see some of these trends, okay? So this was a big deal. And it took a while for researchers to really chase this down and hammer out the mechanistic understanding of socioeconomic status predicts, I don't know, I'm making up numbers, 11% of your Australia-specific risk. Your latitude, or your distance to the sun, radiant energy decreases with the fourth power of the radius from the emitter to the receiver. So location has a very, very strong effect, right? There's a reason that Mercury has no atmosphere and is like thousands of degrees at the surface of the planet. Same thing with Venus, but a little bit cooler because it's a little bit further away, right? So all of these play a role. And as researchers, we can make these educated a priori guesses or at first face a priori looks at it. We can start to put these things together, and then we can test by looking for specific canceration subpopulations in specific areas, in specific socioeconomic classes, in different ethnic and racial groups, and so on and so forth, and look for a much more specific and detailed understanding of what is the association of each and all of these factors. And so when we throw all of these things in together, having researched each of them individually, we can put together a structural equation model of, we've got these 6 boxes. Each of them is an arrow with an associated strength of the output of likelihood and severity of getting skin cancer. There are different kinds. We can say 11% here, 10% here, 12% there, and then add all of those R squares, all those percentage influences up, and say, all right, we've explained 89% of this problem. And of that 89%, 62% is social norms, right? Whatever. Now that other 11%, that distance from 89 to understanding 100% of the behavior of what's going on, we don't know what that other 11% is. Part of it is going to be just sampling error. We didn't ask the right people, or we did a random sample and we just happened to get a bunch of folks who get really bad sunburns in that particular survey. And we don't know what we don't know about that. It could be that we have mischaracterized this. So it's not the ozone hole, but it's a specific event during the opening and then dilation back down, and now it's opening back up again. Folks who were at the beach during that 10-year period more, specifically that time frame, but we can't test for that going back. And nobody has that detailed of a memory of when they were at the beach between 1987 and 1997, right? That's just error. It's going to be in that 11%, and we might not ever be able to figure that part out. But 89% of the mechanism, that's pretty good, right? If you have 89% of the parts in your car engine, it might work. Yeah, close enough for government work. Okay, we got to enter right there. I'm around for questions if you have them. Come back on Thursday. And for Thursday, in your ongoing quest to nail down your literature review, I want you to read an article and prepare to discuss it with us in class. We're going to do a little bit of a journal club. It's a very healthy professional practice. Each read your own article and, give a 30-second, 1 minute, 2 minute explanation of, why do I care about this article, or why don't I care about this article? Okay, cool. We'll see you then.